Good morning, or good afternoon, good evening, depending where you are. And thank you to everyone for taking the time to attend this webinar. It's really a pleasure today to have here as ECE Distinguished Senior Speaker, Dr. Guillaume Baffou. So Dr. Baffou is a research scientist at CNRS, which is the National Scientific Research Center in France, and he works at the Institut Fresnel in Marseille. His research focuses mainly on the interface between nano-optics and small-scale thermal effects, in particular in their interaction between light, plasmonic metallic nanoparticles, and all resulting applications in physics, chemistry, and biology. In 2015, he was awarded the prestigious bronze medal from CNRS, which is given a recognition has been given recognition for his important contribution in the field of thermoplasmonics. And he also authored a book in 2017 on the specific topic. And it's a really good reading. So I suggest it to everyone who's interested to, even if he's already a researcher or he wants to start research in thermoplasmonics, I think it's a great introduction and a great handbook for that. So with that, I leave the word to Guillaume, who's going to talk about thermoplasmonics, heating method nanoparticles using light. Thank you very much. And yeah, Guillaume, whenever you want, you can start. Just a note, if there is something that needs to be clarified, just feel free to type in the Q&A. And if it's something that can wait until the end, then we will take questions at the end of the talk. But if you don't understand something and you feel you need a clarification, just type it down and I will give you the word or I will ask the question to the speaker. Okay? Thank you very much. And yeah, you can start.
Thank you, Alessandro, for this nice, kind introduction and for the invitation. So it's a pleasure today to be here to talk about a field of research that I have been working on for 13 years now, and which consists in heating metal nanoparticles using light. Sunlight, nanoparticles absorb light, they get hot. And this is what I have been most of my research time during this last 13 years. So when I say this, usually people wonder how could such a simple picture occupy one researcher for so long? Well, if you think about it, as soon as you do that, you end up with a nanoscale point-like source of heat. And this is one of the most fundamental concept you can think in nature. So just for this reason, you can investigate, you can do some very fundamental research in nanosciences. And the second reason why you can do a lot of things is, if you think about it, any field of science features some thermal-induced effects. Chemistry, biology, of course, fluid dynamics, magnetism, phase transition, fluid convection, fluid dynamics. So because of this, you can investigate almost any field of science if you are working in thermoplasmonics. So this is what makes the beauty of this field of research. So just to give you an idea of how important this field of research is, here are some statistics regarding the number of papers per year. So as you can see, this field of research was born in the early 2000s, and even now it's still increasing very, very fast. So it's still not the end of thermoplasmonics. It's still a very active field of research today, and the purpose of this presentation will be to explain what's going on, what are all these papers here. So right now, just to give you an idea of the outline of this presentation. So I'm not sure who is here, and if all of you are familiar with plasmonics, so I really wanted to start from scratch, from zero. So I will give you a short introduction of plasmonics. But then the main part of my presentation will be divided in two parts. So one fundamental, to explain you what kind of fundamental research you can do with this point light source of heat. And I will take some example from my research activities these last years. So to tell you what we can do in physics, chemistry, and biology at the nanoscale. And then I will tell you what kind of applied research we can think of, how we can make science advance using this heating of particles using light. So what is plasmonics? So if you consider this gold nanosphere, let's say 20 nanometer big, and you shine light, the electric field of the light will make the electrons, the free electrons, oscillate. And because you have oscillation of the electrons, you have charge accumulation at the boundaries, a bit like in picture C. And this charge accumulation creates an enhanced electric field, which means an enhanced light intensity. So the interest of nanoparticles is that you have a strong enhanced of the light intensity at the vicinity of the particles. For instance, here with this gold nanosphere, you have an increase of a factor of 50 at the vicinity. But this effect occurs at a very specific wavelength range, which is here around 550 nanometers, and this is called the plasmonic resonance. And what makes all the interest of plasmonics is that you can play with this resonance. If you change a bit the geometry, for instance, if you consider a dimer, 2 nanospheres very close together, the charges I mentioned, they are now very close, which creates a huge electric field, which means a huge light intensity. So because of this, in this gap, you can have a light enhancement of 3 orders of magnitude for this system. And you have also a redshift of the resonance. Another way to play with this resonance, this plasmonics, is to modify the shape of the sphere. And if you make it longer, if you elongate the shape, you also end up with a shifted resonance to the red, and an even stronger enhancement of the electric field. And you can even think about combining these 2 effects. And then you end up with a light intensity that is up to 5 orders of magnitude larger than what you could achieve with a laser you have in your lab. So let's say now you have some molecules around. If the molecules are lying in the gap, they will interact with 5 orders of magnitude more photons compared to your simple laser. So this is one of the most important interests of plasmonics. You can play with light at the nanoscale, confine it, and enhance the light intensity. So this is to summarize interest. You can play with light, enhance, and confine, but at the same time, just because you have charge oscillations in a metal, you generate heat. And you have this kind of duality in plasmonics. You can enhance and confine light, but also enhance and confine heat. So you have this double, very fundamental tool, light, which deals with light and heat. So this creates like 2 communities in plasmonics. The one is interested in generating heat, and the second one is interested in confining light. But when you are only interested in lights, you also have to mind heating effects, because it's, let's say you want to shine molecules with plasmonic structures, you don't want to heat them. You can bleach them, you can damage them. So you also have to care about photothermal effects, also if you are working with light. So my message is that today the impression I have is that the whole community, the whole plasmonics community, is concerned with photothermal effects, not only the heat part of the community. So now this was introduction regarding plasmonics. Let's focus on what we can do very fundamentally when we heat nanoparticles on the nanoscale. So this is a picture, a schematic, explaining what I have been doing this last 10 years in thermoplasmonics. My typical experiment was a carpet of gold nanoparticles. I shine a laser, I focus a laser on that, and the effect is to increase locally the temperature. So is it challenging to do this? Absolutely not. It's very easy if you can afford buying a laser and gold nanoparticles, which is not that expensive. You shine the lights on the particles and you heat them. So this is not complicated. This is not a challenge. The challenge, if you want to do proper science, is more to measure the temperature, to know the temperature. This is complicated because this is occurring on the microscale, on the nanoscale. So to measure the temperature, the community has to develop some tricks. And here you have an overview of all the temperature I'm sorry, the title is in French. This is thermal microscopies. You have a list of all the temperature microscopy techniques that have been invented for plasmonics. And as you can see, they date from 2009, which is quite late. Thermoplasmonics is much older than that. And most of them, they are based on the use of fluorescent molecules. And the idea is to disperse, to scatter some fluorescent molecules in this kind of sample. And just because fluorescence is temperature dependent, if you map fluorescence, you map the temperature. This is not more complicated than this. But the problem is that fluorescent molecules, they can be sensitive to light, they can photobleach, thermobleach. So there are some artifacts, and then it's invasive. Sometimes you cannot put fluorescent molecules in your system. So we developed a technique which is a line named, each dot is one paper. So the line named wavefront sensing is a technique we developed in 2012, and this technique is label-free. You don't need any molecule around to map the temperature. You are just mapping the wavefront distortion due to the thermal lens effect. If you consider this system where you have liquid on top, substrate below, and you shine laser lights to heat this particle, you're gonna change the refractive index of the liquid, and this will create a thermal lens effect. And this technique we developed consists in measuring this wavefront distortion, and from this wavefront distortion, we retrieve the temperature. And the interest, again, is that it is label-free. We don't have to modify our system. And this is the wavefront distortion we measure when we heat. And from this wavefront distortion, which I am reproducing here, figure B, we can retrieve, using an algorithm, the temperature map and the heat source density, these two maps. And this technique is not only working with single nanoparticles, but for any system that is heating on the microscale, we can retrieve the temperature gradient. For instance, here, if you illuminate many nanoparticles at a time, it's working as well. So thanks to this very convenient temperature microscopy technique that is label-free, you don't have to modify your system, we could apply this temperature microscopy technique for many applications in physics, chemistry, and biology. And this is what I'm going to explain now. So again, this is one the typical approach we had this last 10 years, shining many nanoparticles with a beam, the laser beam. So this is a side view of the experiment, laser from below, gold nanoparticles, we heat the liquid. And this is one of the first images we obtained using this new temperature microscopy technique. So I don't know how surprised you are when you see that. We were very surprised when we discovered this first image. What is surprising here is that we can achieve 200 degrees in the sample. But as you can see, we are in the liquid, we are in water, so at some point we expect a bubble. At least at 100 degrees we should see a bubble, but we didn't. At the beginning we thought that we had a problem with the algorithm. Maybe we are missing a factor of 2 in the code or something, but it was not the case. We did achieve 200 degrees in water in this kind of sample without boiling the fluid. And the reason of this observation is that you should not boil water at 100 degrees, actually. This should not happen. The reason why it's happening in your kitchen when you cook a star or anything is that because you have nucleation points in the pan, in the vessel you are using, which means scratches, roughness, microscale roughness, dust, a piece of dust, and so on. In this kind of experiment, and in many experiments in plasmonics based on glass substrates, there is nothing flatter than glass. So we don't have nucleation point in this experiment, and this is why we don't have bubble formation. We do have bubble formation, but close to the spinodal temperature, which is close to 200 degrees. So above 220, yes, we see bubbles, but not below this temperature threshold. So this is this kind of new physics you can discover in thermoplasmonics when you hit at the nanoscale, as an example in physics. But then we thought, okay, this is nice to evidence. We could tell the community that when they see bubbles, they don't have the temperature they expect. But is there any application of being able to heat a fluid, to heat water at 200 degrees? Well, the answer is yes, in chemistry. Chemists, they are using liquid water at 200 degrees as a solvent. So we thought maybe we could invent a new concept in chemistry, which could be hydrothermal chemistry, but at ambient pressure. Because chemists, when they want to do hydrothermal chemistry, which is chemistry at 200 degrees in water, they use these autoclaves, which are pressured chambers that enable heating water above the boiling point. But this is bulky, this is, you have a lot of constraints using this. We thought maybe using this kind of sample, like a micro dish with gold particles at the bottom, could be much easier for the community. So we tried. We tried to find a chemical reaction, and this one was great because it creates crystals, and crystals are easy to spot in a substrate. And this reaction creates indium hydroxide crystals. So we tried with this reactants, and it worked well. We did manage to form these cubic crystals on the microscale, which was the evidence that we could superheat the fluid. I'm just reading the question. I will answer it. So it was evidence that we could superheat the fluid, but also it also show a new concept in chemistry, which is doing this kind of experiment on the microscale, but with a temperature gradient. Here on the right, you have the temperature gradient created by the laser beam. And the interest of doing this is that we evidence temperature thresholds, above which we have a different crystallography. For instance, at some point we have these pine tree shapes, and below a given temperature we have the cubic shape. As a chemist, if I wanted to investigate this question, I would have to use many autoclaves with many different temperature to reproduce this experiment. Here, in a few minutes, with a single experiment, we have the answer of the question here. So this was the interest we evidence of this new approach. So of course, this approach is not meant to produce industrial amounts of products. It's not the aim. This approach we have introduced here was more for fundamental research. So the question from Steve Aram was, if the glass surface is not very flat, or if it has scratches, would it start bubbling at lower temperatures? The answer is yes. If you try to do some, we never tried, but we expect this. If you try to do some thermoplasmonics right on the scratch, if you scratch with a diamond tip, for instance, on glass, I'm sure we will see bubble formation. So yes, this is a message we convey in this paper, yes. But what was surprising a bit is that these nanoparticles, these gold nanoparticles, do not act as nucleation points. We could have thought that, but it's not the case. So what I believe is that a nucleation point should be rather like a micrometric cavity to enable like a first bubble with a flat curvature, rather than nanometric bumps. So I hope I answered this question. So far I gave you some example of what we can do in physics and chemistry, and the last example I would like to show you regarding fundamental applications is in biology. What we wanted to do lately is to heat single living cells. The sample is exactly the same, a carpet of gold nanoparticles and some living cells cultured on top. So we wanted to heat them one by by one to investigate the effect of temperatures on cells. So this is a full field of research because cells are highly temperature dependent. But usually when biologists want to do that, they heat the whole stage, the holder of the sample, or even the whole microscope to 37, 38, 39, 40 to check what the effect of the temperature is. But when you do that, you have a huge inertia. You have to wait maybe 10 minutes, 15 minutes before reaching the target, temperature target, which means that this is fine. You can still see the effect of temperature, but you cannot investigate the dynamics because cells, they are responding very fast. So you need to investigate the fast dynamics, the fast response of cells. You have to be fast, and to be fast, you have to diminish the thermal inertia. And to diminish the thermal inertia, you have to heat at small scale. And this was the aim here. When you do that, what is represented here, you can heat and achieve the steady state in a few milliseconds. So you have the time zero to investigate any temperature-induced effect in living cells. And what we could do is to map at the same time fluorescence arising from the cells and temperature without any problem, because the temperature microscopy I presented is not based on fluorescence. So when you use this temperature microscopy technique, you are free to use any fluorophore you want to label your living cells. So this is what we did, and when biologists want to investigate the heat shock response, something I should explain before explaining the results. When a cell is stressed, and especially when it is heated, it's very bad news for the proteins that tend to unfold and lose their functions. But to fight this problem, nature invented a trick to solve this problem. And when a cell is stressed, there is the expression of proteins, which are called heat shock proteins, and which are meant to protect other proteins, to avoid unfolding of the other proteins. And one way to follow this expression of proteins is fluorescence. So I won't enter the details about how to label the cells, but when the cell is fighting against temperature increase, you can see it by fluorescence with the appearance of granules, hot, bright spots in the nucleus of the cell. So let me answer this question. Does the cell distort the wavefront as well and affect your temperature distribution? So here, this is a good question. The second image you have in the middle, it's a temperature map arising from the wavefront distortion created by the temperature gradient. You remember that? So it is true, we have a living cell that is also distorting a lot of the wavefronts arriving on the sample. So what we do to discard this effect is that we take a reference without heating, but with the living cell. And then we take another wavefront image upon heating. And then we subtract the two images to get rid of the effect of the cell. And we end up with only the effect of the temperature. So this is how we get rid of this effect. So it's not perfect. As you can see on this image, you still can distinguish a bit the cell. So this is because this subtraction is not perfect. This is maybe because the cell is moving between the reference and the image. But this gives still a very good idea of the temperature increase if we do this. So now let's go to the fluorescence image. So at the bottom, you have laser on, laser off. And when it is on, you see the appearance of the granules in the nucleus. And when it's off, they disappear, and it's reversible. So here, the cell is trying to fight against the temperature increase by producing these heat shock proteins. So it's working well. We managed to reproduce what biologists have done by heating the stage of the microscope. We could do that, but with a much faster dynamics. And this is what I want to show in this slide. On the right, you have fluorescence intensity as a function of time for the two granules, 1 and 2, that appear in the nucleus. And as you can see, so we have been heating for only 1 minute, but even after a few seconds, already you have the appearance of the granule and the response of the cell. So the cell reacts very fast when it is stressed. And this is something you can realize only when you heat very fast. And thanks to this microscale approach, we have the time zero because we can heat very fast. So this is the benefit we evidence with this paper. And the other benefit that is also a very important benefit is that, let's say you heat too much and you kill your cell, you don't have to take a new sample. You just move away, try to find another area in the micro dish, heat it at 37, 38, another temperature, and when you are done with this area, go to another area, heat to another temperature, and you can spend the whole day on a single micro dish and investigate many temperatures with a single micro dish. This is not possible when you are heating your stage. You have to take one micro dish per temperature. And this is highly sample consuming, time consuming. So this is a new way of doing thermal biology at the level of a single cell. And I am expecting this field of research, that is single cell thermal biology, to be rising in the next years. So this was the part regarding what we can do fundamental in thermoplasmonics in fields as various as physics, chemistry, and biology. So now let's focus on research activities that are trying to make science advance, really applied research. So what can thermoplasmonics be for? So the most famous application of thermoplasmonics is plasmonic photothermal therapy. You can cure cancer by heating gold nanoparticles. And the idea is very simple. It dates from the early 2000s. It's actually an idea that came from Rice University. So I'm talking about it, but honestly, Rice University is the best place to hear about this activity. So the idea is very simple. You put gold nanoparticles in a tumor. To do that nicely, you inject it intravenously, and these particles, if they are functionalized with proper antibodies, they will attach to cancer cells and not to healthy cells. This way, after some time, all the particles, or most of them, or some of them, they end up in the tumor. You shine light, and you burn the tumor, and only the tumor, because gold particles are only located in the tumor and not in the healthy tissue. So, explained like that, it seems very simple, very trivial, but the idea dates from 2000. So 20 years after, I'm not still able to tell you it's working, it's widely disseminated in the whole planet. From what I know, but again, Rice University is the best place to know more about this. From what I know, so there is a company working on this. Sorry. Yes, I have this slide to show you. So this is a schematic, and this is what's really happened this last 20 years. So first, preclinical experiments on mice with this usual subcutaneous tumor on mice. So people inject gold particles, shine laser, and after some time, days of treatment, indeed, they make the tumor disappear on mice. So this was 2008, and the paper published in 2008 was impending clinical impact. So we were expecting a very near impact of the technology, but actually it's only now that, from what I know, many patients have been recruited to do this clinical trial. So I still cannot tell you if yes, it is working or no, it won't work. I think we have to wait 1 or 2 years more to really have statistically relevant results regarding this application of plasmonic photothermal therapy. But there are some problems. So just to answer the question of Sivaram, interesting temperature profiles in the cell. Do these white spots, this is regarding the last part. So the bright spot we have seen the nucleus, they originate from DNA. These are the location in the DNA that correspond to the genes that code for the heat shock proteins. So yes, so far the clinical trials, they are based on cancer of prostate cancer. But the problem in this field of research, as you may expect, the body is not transparent. And this is one of the main problems you have to face with when you investigate this kind of application. And to fix this kind of problem, so researchers from Rice University, and especially Naomi Halas, the idea is to use an optical fiber that is put directly inside the tumor to directly drive the light within the tumor location. This way we don't suffer from the problem of the non-transparency of human tissue. So I don't know much about the results. What I know is that the last news I got from this kind of application was in 2018. I could listen to Dr. Halas in the Gold Conference in Paris. And at that point, they tested this technology on a few patients. And the result, the outcome was that, so she said that one of the patients could ride a bike after the treatment. So I don't know if it is good or bad, but I think it's good. So everyone was very excited by this. So I was a bit skeptical because when I can read papers from the literature, usually this is not statistically relevant. When you have to make sure that a technology, a biomedical application is working, you have to conduct experiments on many patients. And at the end, we can say that life was make longer by a few months, few years, and so on. So maybe it was too early to motivate the community regarding that. So I hope it will be working soon, but the answer should come up, at least I think, from what I can see from the website of Nanospectra Biosciences in 1 year or 2 years. So this is what I can say from my side, from what I know. But again, Rice University is the best place to know everything you should know on this kind of, on this precise application. The second very active application of thermoplasmonics concern solar light absorption. More and more applications are based on the fact that solar light is free. It makes sense to try and get it. And since plasmonic particles, they are enhancing a lot light absorption, it makes sense to try and harvest solar light using plasmonic nanoparticles. So this is a basic idea, and many applications, I mean, some applications have been thought, like water desalination, thermophotovoltaics, vapor generation, medical tool sterilization, solar energy storage. I won't detail them all. I don't have time to do that. I will consider them as a whole, because all of them, they are based on the same principle, absorbing as much solar light as possible. So one typical experiment to absorb, one of the very first basic experiments to absorb solar light was this kind of textbook case where you have a solution of gold nanoparticles in a tube, and you have a solar light, and the aim is to focus as much light as we can using a Fresnel lens, as you can see here. And this way, in this publication, the researchers were able to heat and even boil water. So, so I like this paper because it's a perfect example of a misconception that sometimes is found in the literature regarding the fact that when you illuminate many particles, you do not necessarily have localized hotspots around each nanoparticle. And this is a now famous photothermal collective effect in plasmonics, and I would like to explain this effect because it's now very important in this field of research. So if we focus on the abstract, solar illumination of nanoparticles in a liquid produces vapor without the requirement of heating the fluid volume. So they say they have vapor, even boiling, but they don't heat the fluid. So for me, this cannot happen in this kind of experiment. So I tell it I don't agree with the statement here. And in particular, with a paragraph here, so this is the common misconception I'm talking about. So this expression here is a temperature increase, is the expression you have to use to calculate, to estimate the temperature increase you get when you shine light on a single nanoparticle. So P is the absorbed energy by the particle, R is the radius, and K is the thermal conductivity of the surroundings. So in this publication, the researchers were surprised that when they use this expression, they end up with a temperature increase of 0.04 degrees, which is a drastic disagreement with the experiment where they see boiling or vapor generation. So my message is that, of course, you end up with 0.04 temperature increase with this expression because it's considering only one nanoparticle. And here, many nanoparticles are eliminated at the same time. And I would even say here, you shine light on the black solution. And this expression considers that you have a transparent solution with a single nanoparticle in the middle. So of course, in the second case, you don't heat, while when you have a black solution, you do heat. So this is why this expression should not be used when you eliminate many nanoparticles at the same time, because obviously you end up with an underestimated temperature increase and a false counterintuitive problem. So this is a famous photothermal collective effect often encountered in plasmonics, which led to many wrong discussions in papers, and I can understand that because this is highly counterintuitive. When you shine, so this is another example taken from the literature, when you have this very sparse density of nanoparticles, called nanoparticles, and these are numerical simulations regarding to this experimental sample. So in shine light, you have hotspots, you have confined temperature increase around each nanoparticle. But this is only when the laser beam or the light beam is on the size of the field of view, which is 1 micrometer square big. Now let's say that you increase the beam size, but with the same intensity. You don't vary the same intensity, only the beam size. Then this is a new temperature profile. And as you can see, we lose completely the hotspots. Now the temperature is perfectly uniform, first. And second, the temperature increase is much higher than what you have when you have a small beam size. So my message is, when you shine many nanoparticles at the same time, very often the temperature is uniform, you don't have hotspots anymore, and it's much larger than what you can achieve with this temperature increase here. So why is it so counterintuitive in plasmonics? That's because this kind of image I have shown at the beginning, this kind of near-field maps, that do not depend on how many nanoparticles are in your sample. If they are far enough, further enough from each other, you are safe. You can safely state that this near-field enhancement profile, they are true. No matter how many nanoparticles you shine, only the interdistance matters. This rule of thumb is not true in thermoplasmonics. In this case, not only the interdistant matters, but also how many nanoparticles you shine. So to know if you have hotspot like that, you have to mind the nanoparticle size, the beam size, and the interdistance. So there is no counterpart in optics regarding these photothermal collective effects in plasmonics. So this is counterintuitive for people working in optics, and this is why naturally you don't think about this effect could occur in plasmonics when you are working in plasmonics. So let's go back to this solar light absorption. If this is the temperature profile we get with no hotspots, the natural question is, so what is the interest of using nanoparticles then? If I paint my sample with black paint, I would get the same temperature profile. So what's the point using gold, using nanoparticles for such such an application involving wide wavelengths, wide range, solar range absorption, and macroscopic heating. So for all this application, water desalination, photovoltaics, vapor generation, medical tool sterilization, and solar energy storage, all of them, they are based on heating a big object, like a macroscopic area, and using solar light. So we can wonder if it is really necessary, useful to use gold nanoparticles and not black paint. So what one can answer, of course, using plasmonic nanoparticles is better because you benefit from a resonant absorption. This is true, but believe me or not, but even with plasmonic, so black paint means 100% absorption. And even with a plasmonic resonance, you will never ever achieve more than 100% absorption. So gold nanoparticles, they will never do better than black paint. So what is the aim then? Why would we want sometimes to use gold nanoparticles instead of black paint or carbon black for thermoplasmonic applications? So this is an important question. And I find the answer lately, actually. It's not that I mean, the answer is simple, but not that simple to find, I guess. So to answer this question, let's go back to the experiment I have presented. Here, if I paint my glass cover slip with black paint, would it work? So the answer is yes, it will work. I will manage to heat my cell, and it will certainly act as if it was acting with the gold nanoparticles. But still, here we did use nanoparticles because if we use black paint, we cannot see anything anymore. We cannot collect fluorescence anymore because the sample will be dark black. So here we wanted a substrate that was absorbing red, but transparent in the visible range, transparent for the blue and for the green. So gold nanoparticles, they make sense when you want, I think, to absorb only parts of the spectrum and not the other parts. But when you have sunlight, the spectrum is very broad. So you don't really need a resonance to absorb a very small part of the spectrum. So my conclusion, what I can propose is that using nanoparticles makes sense when you want a spatial confinement, when you want to heat on the nanoscale. Black paint won't do that. When you want a temporal confinement, for instance, at the millisecond, nanosecond, femtosecond timescale, or the nanosecond timescale, then only a nanoparticle can do that. Or when, this is what I just said, when you want a spectral confinement, when you want to absorb light over a restricted range of the spectrum, then you have to benefit from a resonance. And for this, nanoparticles, gold nanoparticles are better than molecules, for instance, because molecules, they can photobleach, thermobleach, and gold particles, they are very stable with temperature and light. So this is what I can propose for the interest of using nanoparticles compared to black paint and carbon black. So this discussion I just proposed here, we are discussing that, we are telling that in a very recent accepted paper, which is still not published, with my colleagues Frank Stichos and Romain Kikidon. And the last practical application I wanted to discuss, this is again a highly debated field of research. This is why I wanted to give my opinion regarding this field. It consists in driving chemistry using gold nanoparticles. So what researchers have seen a bit more than 10 years ago was that when we introduce some gold nanoparticles in chemical reactions, the chemical reactions can be favored, enhanced. The chemical rate can be enhanced as soon as you put gold nanoparticles in the sample. And some people, they say, this can be due to a very fancy process where electrons from the particles, some hot electrons generated from a photon absorption, these very energetic electrons, they can participate and be injected in surrounding molecules. to generate, for instance, here the oxidation of an ethylene molecule. This is what was evidenced in this Nature Chemistry paper in 2011. So this was the beginning of the story. Yes, and this is what they measure: the rate enhancement as a function of time when light is on and off. And indeed, this is very impressive. There is a huge increase of the rate enhancement as soon as the light is on, if there are nanoparticles in the sample. So the main problem with this field of research, and which is now a very general problem, well-known problem, is that what about thermal effects? This very reaction we have here can be also enhanced. This is a shame, but it is like this, just by heating. If you heat, you have the same enhancement. So what if these researchers were not just heating their sample with light? So you may say they should have thought about that. And they did actually. They did, and they checked that. So unfortunately, they didn't use an infrared camera, which would have been great to check the temperature. They use a quite indirect technique, but which looks good. They say, let's vary the power of the laser. Since it is a chemical reaction, and since we expect the Arrhenius law, if we increase the temperature, we should observe an exponential increase of the chemical rate, which is true, and which they did not observe. You see, it's perfectly linear as a function of the temperature, which is as a function of the, at least as a function of the light intensity. But I investigate a bit more these results, and what I have seen is that if you try to fit this set of data with this function, which is the Arrhenius law, this is what you find. So you see, unfortunately, so this was a good idea to check for photothermal effects, but unfortunately, they didn't span a sufficiently large light intensity range. Oops. It is much too small to evidence whether we have or not a temperature effect. So this is one example of a paper that is not convincing enough for the literature to make sure that there is no photothermal effects in this paper. So this was very early. This paper that's from 2011. At that time, people were not really aware of this problem in the community. But now more and more papers are published on this topic. Sometimes they are very, let's say, some of them, they are even aggressive. But so I'm not sure we have to be aggressive in research, but the benefit of it is that because of this researcher, which are a bit pushy regarding their ideas, now everyone is aware of this effect in plasmonics chemistry, and everyone is trying to solve this problem. So being aggressive can be bad, but at least it makes science advance. And so in this case, in this paper, we try not to be aggressive. On the contrary, the idea of this paper to help this community was to give some tricks, procedural, so it's very recent, it has been accepted a few weeks ago, give some tricks to evidence temperature increase in experiments in plasmonics, because as I said, it can be complicated to know whether you have or not a temperature increase in your plasmonics experiment. So I won't detail the 7 tricks we have proposed in this paper. Feel free to read them if you wish. I will just comment the first figure of the paper, which is reviewing all the papers that have been published these last years and that are focusing on this problem, whether photothermal effects can explain the rate enhancement in this hot carrier assisted plasmonic chemistry community. And you see, it's impressive. Now, almost every month, you have a new paper coming out saying whether yes or no, there are some photothermal effects or not in this field of research. And I have a question for the audience now, so I guess you can now use your question and answer window to answer this question. What do you think are these blue points? There are also many of them, also related to this field of research. So these are not the brown points, but there are many as well regarding this field of research. So I don't know if you have some ideas. So the answer is all these blue points, there are review papers on this field. So this is very surprising. At least I feel surprised by this, but I can see it every month, not months, but every 6 months, a new review paper is released on this very topic of hot carrier assisted plasmonic chemistry, which is a lot. I'm not saying that all of them say exactly the same thing. Of course, any researcher has his own sensitivity and explains things differently, but maybe this is too much. And I think this is a trend today to publish review papers. Journals, they like it because they are highly cited. Researchers, they like it because it's highly cited as well. But then it creates some noise on the field, I think, because a review paper is here to make clear the field of research and to avoid people reading many papers before having a clear idea of the field. But here, because of these many review papers, you have to read many papers to have a clear idea of the field. So soon we'll have maybe reviews of reviews to clarify a bit of this field of research. I don't know. But this field of hot carrier plasmonics is really an example where you have this over publication of reviews, and I think it's a shame for the field. Yeah, reviews on hot carrier assisted plasmonics chemistry. So just to summarize, to go back to my previous slide, that was like a landscape of the field of plasmonics today. So on the one side, you have the heat community. On the other side, you have the light community. Now also the light community is concerned with photothermal effect because they don't want to heat, but to make sure they don't want to heat, they have to measure the temperature. So now the full field of plasmonics is also working in thermoplasmonics, I think. But now you have also this third part, which is hot electrons. So this is why you have a very active and passionate field of research in plasmonic working in this field, because it is a brand new playground for the field. I'm not saying that we are bored with light and heat, but this is new, and as it is new, we can understand that a lot of activity is around this topic. I just want to say that this topic is highly debated, as I said. Some researchers even think that it's not possible to drive some chemistry with hot electrons, mainly because of the very short lifetime. This is not the majority of the community. Most of the researchers, they think we are able to do that. Most of the community now thinks that you have this dual effect, heat and hot electrons at the same time. But now I think this is a landscape of plasmonics, light, heat, and hot electrons. So since 10 years ago now, the landscape of plasmonics is enriched with this hot electrons thematic, but still all these three fields, they are concerned with photothermal effects, with thermoplasmonics. So thank you for your attention. These are the people I have been working with during these last years and the financial support. And before finishing this presentation, I just want to advertise this spring school plasmonics summer school on thermoplasmonics. So this is the first time I advertise this school, which will be scheduled in June 2021. So for the moment, just save the date. So this is the incomplete list of invited speakers for this conference occurring in 1 year. And thank you all for your attention.
Thank you very much. Yeah, it was a great talk, and it's very interesting to see how active are some topics of like the highly debated research fields that you underline. I think it's really useful to kind of discuss about what science thinks and what are the open problems that still need to be solved. So let me give the word to Sivaram, so that he had another question. Okay, so you can speak directly.
So it's a very nice talk. I enjoyed it. It's very clear that there is overlap of thermal and optical effects that will bring interesting observations and possibilities. So one of the things that you forgot to mention, you did mention confinement, spectral confinement, spatial confinement, and temporal confinement. That's fantastic. But nanoparticles do provide a way of, delivery of, for example, drugs and so on. So I just want you to comment on that, if that's okay.
Yeah, I think, your comment goes in the spatial confinement, I guess. Well, actually, you are right. Gold nanoparticles, they make sense when you want to work in biomedical application with something in your blood also. This may be actually a fourth line of the interest of using gold nanoparticles, because of course you won't put black paint in your blood. So yes, so you're right. For biomedical application, in particular drug delivery applications, of course black paint cannot be used. Yes.
And so Julian, so you typed the question, but maybe you can just repeat it if you want.
Hello, thanks for the interesting talk. Yeah, my question was regarding the wavefront distortion technique, and what is the previous knowledge that you need from the system? Do you require knowing the thermal dependence of the refractive index?
Yes.
And in that case, how do you consider that you have water and glass?
So what we need to know is the dN over dt, how the refractive index is varying as a function of the temperature. But this is the only parameter we need to know. For instance, we do not need to know the thermal conductivity of the substrate of the liquid on top. We need to know it only when we want to compute the heat source density, which is this image. Oops, sorry. Here. In this case, we need to know the thermal conductivities of the surrounding, but for the temperature, we only need to know the dN over dt. And the assumption we made is that we have an infinite water medium on top and an infinite glass medium below. We have infinite medium on top and below to have a very simple green function. To go from this wavefront distortion to this temperature map, we have to use an deconvolution algorithm based on the use of a green function. And the green function is simple if you have this infinite media on top and below.
Would it be more complex for different geometries?
Yeah, more complex. Sometimes we are using a thin layer of liquid, like a sandwich, like glass, liquid, and glass. In this case, you need to know the thickness of the liquid to compute what should be the new green function. If one day we decide to work with a sample substrate that is not flat, then it won't work because it will be very complicated and the green function won't be simple. So it works with very nice geometry and typically with a glass cover slip with a liquid on top.
Thank you very much.
Thank you, Guillaume. So other questions here, especially from students? I mean, if you have a question about this, it's really an opportunity for you to talk to one of the top experts in this field. So I would think about asking something. In the meantime, so yeah, I had a question when, so you mentioned at some point photothermal therapy for cancer, or in any case, every time you need to disperse nanoparticles in a volume. So many of the works, and of course, many also of your works focus on the array of 2D nanoparticles. Do you think there is some some intuitive or non-intuitive idea when you move to 3D arrays or distribution of nanoparticles? Is there something you have to keep into consideration, or is just an extension when you have more heaters?
You mean why sometimes we are using a 2D distribution of nanoparticles and why sometimes we prefer 3D?
No, I mean, sometimes you need 3D, like if you inject particles in a tumor, that's what you end up with. I'm just saying from the fundamental point of view, if there is any particular idea or particular thing you have to pay attention to when you have then to calculate, when you have to predict the temperature distribution in that case.
Well, the only problem you may face is this photothermal collective effect, which happens when you have too many nanoparticles heated at the same time, like in this picture. So this effect occurs in 2 dimensions, which is the case in this cover slip, and it's even which can occur even with a much higher probability in three dimensions. The rule of thumb is that when you have a liquid with nanoparticles, all the time you have this photothermal collective effects. All the time you have this very uniform temperature increase in the solvent, in the liquid, and an enhanced temperature increase compared to what this expression can give. So in 2 dimensions, maybe you don't have this photothermal collective effects, but in 3 dimensions in solution, you will never have this localized heating effect, this localized hotspots.
Right. But you will still have, like, in the sense of geometry, meaning that if you have like dispersed nanoparticles above a certain concentration in a volume, and you are absorbing solar light or a laser, you will still have confinement in 2D, meaning that light wouldn't be able to penetrate much deep into your flags. So in that sense, you might have confinement, not hotspots, but vertical confinement.
Yes.
Yeah, that's all right. So we passed 11. So if there are no other questions, let's see, no one. Okay. Okay. So again, I would like to thank Guillaume for his great talk, very stimulating, and would like to thank all of the attendees for taking the time to listen to the talk, and hopefully I'm gonna see many, if not all of you, at the next seminar in August, and we will advertise it. And well, thank you very much to everyone, and have a nice day.
Thank you all. Bye-bye.